Awesome. So appreciate everybody showing back up after lunch. It's kind of a cruel thing to put a bunch of geneticists on the docket right after everybody sat in the sun and eaten a whole bunch, but we'll try and keep things as interesting as possible. I took this opportunity to give sort of a really broad picture survey of where we're at with genetic approaches, particularly on the beef side. There are a bunch of folks that are more competent to talk about the dairy side of things, which is a little bit further along than we are in the beef sector. But things that are working well, things that we are still very much obstacles for us, and then a couple of the areas that my lab is working on at the University of Tennessee. All right, so the good news, right, is that methane emission appears to be heritable, right? So as any good quantitative geneticist would tell you, you can't make selection on a trait that isn't passed down genetically. Methane appears to be, at least in grazing beef cattle, and beef cattle that are in feedlots, somewhere in the neighborhood of 0.2 to 0.3 heritability. This is similar to a trait like weaning weight, very important economic trait for beef producers. And so that's encouraging. That tells us that there's ample genetic variation for which we can make selection on and drive the genetic trend of this trait of methane emission forward. Methane emissions also appear to have favorable correlations, genetic correlations with feed efficiency. So we're not going to be breeding productivity out. It was mentioned earlier, the long-term work that's happened in New Zealand on that sheep selection project. Those sheep are no less productive than their high methane emitting counterparts. Very encouraging for us because beef producers, just like all farmers, don't like to lose out on profitability at the sake of something where the economics are still a little bit fuzzy. And the real good news, I guess, for us in the beef industry is that we're continuing to make rapid genetic progress in productivity traits. So this is not necessarily absolute methane reduction, but it's certainly chipping away at our productivity, or at our intensity, rather, of emissions through that continual increase in growth and productivity. I'll talk about this a little bit tomorrow in the beef section more specifically, but in particular on these cow-centric traits, understanding fertility, longevity, the importance that those play on the methane intensity scale. And then beef on dairy, of course, is the big elephant in the room when I'm talking to beef producers. This has replaced a big chunk of our beef production as the national cow herd is at the lowest that it's been since pre-1960 almost. So this is some of the good news with regards to methane emissions. But our big challenge, and I'm sure this will get reiterated multiple times in this genetics section, is that capturing phenotypes is still cost prohibitive and difficult, particularly as we think about a beef industry, a forage-based beef industry. So if I want to measure these phenotypes on a grazing cow, which is the space that our lab works in, very difficult because we don't have those regular touch points in an extensive grazing environment that we get in a dairy. In a lot of cases, we maybe lay hands on these cows 2 times a year as we get out into grazing public lands or in the swamps down in Florida, right? So this is a major challenge. And there's lots of little groups of us that are generating phenotypes, but data sharing and integration, particularly integration into genetic evaluations, has not happened as of yet. But the good news, right, is that if we can measure a trait, we can make genetic predictions, right? That's the business that we work in, are making these expected progeny differences. So genetic predictions about an animal's genes that they can pass down to their offspring. And with those predictions, we can make genetic progress faster. And this is a quote that I use all the time from Professor Mike Coffey at Scotland's Rural College. In the age of genotyping, phenotype is king. All right, genotyping uptake has happened fairly rapidly in the last decade or so in the beef industry. The American Angus Association, which is where this figure comes from below, is genotyping somewhere in the neighborhood of 150,000 animals a year. So we're genotyping lots of animals, but the one thing that stands in our way of making reliable genetic predictions is capturing sufficient numbers of phenotypes on these genotyped animals. And I show this to producers all the time as well. There's this phenotypic paradox, right, where if we're really easily able to measure a phenotype, we get lots of them. So birth weights, all you need is a hand scale and a little bit of courage to go out and grab a newborn calf, and we can capture a birth weight phenotype. Weaning weight requires that you have a scale, but these are by far the most plentiful records that are in genetic evaluations. But as we move further down the value chain, so the further away that we get from the person that made that initial breeding decision things like carcass traits, we have relatively few records. The genetic predictions are slightly less reliable than what we get for something like a birth weight or a weaning weight. And then as we get into feed efficiency, another big thing that we're interested in, the more expensive these phenotypes are to collect, and when there's not an incentive to collect them at the level of the breed association that runs these genetic evaluations, it gets even trickier. So these other traits, animal disease, greenhouse gas emissions, which is what we're all here discussing today, and maybe even things like direct measures of a cow's metabolism or her ability to deal with environmental stress. This is the area that we work in. We're trying to find ways that we can easily, cheaply measure these with technology, or as I'll show in a second, with omics technologies. Can we find indicators for these traits that we know are economically important, environmentally important, have a welfare importance, and can we measure and predict these traits more accurately? And so this is the big work that we're doing right now is in a forage-based system where we have relatively few touch points with these cows, where maybe even a green feed pasture system at the scale of a national genetic evaluation isn't something that's reliable and cost-effective enough for a producer to use. How can we find valuable indicators that would enable us to generate these genetic predictions for methane emission? And so this is a little bit off in formatting, but we're very interested in integrating these precision livestock technologies. All of this happening in sort of a field lab system. So one of our university herds that's got somewhere in the neighborhood of 500 cows that we're getting green feed phenotypes on, water intake, real-time, daily interactions with these animals and these precision devices. Working with Ceres out of Australia on their wearable monitoring technologies, but most importantly, looking at these molecular phenotypes that we might utilize to stand in as proxies in genetic evaluations for things like methane emission or for metabolic efficiency in cows. And the one other piece I'll mention that we're working on is can we continue to ignore the microbiome? I'm far from a rumen microbiologist, but I understand the importance and the interplay between animal genetics, so the additive genetics that we pass down from generation to generation, and the composition of that rumen microbiome, which we know is very, very important in methane emissions. And so I do a lot of work with Dr. Phil Meyer at the University of Tennessee, who's rumen tubing this steer. And my first thing is, how are we ever going to take the rumen microbiome, assaying the rumen microbiome, to a producer? Because this 3-person, 10-minute-long process is not going to cut it in a feedlot processing facility, even in a cow-calf operation. This is not something that we're going to do with any sort of routine, in any sort of routine way. And so we're working with a company called Gencove to utilize a buccal swabbing approach that serves a twofold purpose. The first purpose is it gets us some sort of a proxy for the contents of that rumen, right? It's not going to be perfect, but those regurgitated rumen contents are going to stick around in that animal's mouth. We can capture that on a swab, and at the same time get our DNA sample for genotyping purposes, right? And so the idea being we send this off, we do sequencing on this sample, utilizing sort of a low-pass whole genome sequencing approach. That gives us sort of a 2-for-1 in this sample, right? We get an animal's genotype and some sort of a proxy. We're trying to figure out right now just how good of a proxy that is for the fluid component of that animal's rumen microbiome. And then as we think about how we utilize this, it's moving from just looking at an animal's genetics and how it relates to a phenotype, but trying to actually predict that phenotype, because that microbiome is going to give us a better picture of that animal's environment from a diet, from a stress, from a whatever standpoint, and hopefully help us deliver better predictions of individual animal phenotypes and facilitate some sort of a precision management. And then the final thing that we're doing, utilizing molecular intermediates as phenotypes that we might be able to use to predict things like methane emission in a genetic evaluation, is can we use these proxy phenotypes for hard-to-measure traits? Our friends in the dairy industry have the mid-infrared testing that they do on milk samples. Those are decent proxy traits that would allow us to say something potentially about that cow's methane emissions. We don't have the benefit of doing that in the beef industry, so we've turned to a cost-effective approach to generating RNA sequencing. So this is a 3' RNA sequencing approach that we're down to about $10 a sample. So this is something that we're utilizing these high-dimensional phenotypes and trying to refine down proxies that we could use in a genetic evaluation in the absence of a true methane emission phenotype on that animal. So I think that's all that I have, but look forward to the rest of the panel here.